Good afternoon, everybody. It's really great to see a small crowd here. First of all, I would like to acknowledge the traditional owners of the land we meet, the Yugara people, and recognize their past, present, and emerging leaders. Second, I would like to thank Ranjeny and Jake for putting with the teams this meeting together. And I hope there will be a productive, constructive discussion, because I'm very confident some of the established concepts will be challenged. My name is Gerald Holtmann. I'm the director of the Department of Gastroenterology and Hepatology here. And we developed in recent years with the whole team quite a bit of interest in this field. You just could. But obviously, all of us have choices. You have chosen to be here, and most of you have chosen to take a back seat. And I hope you have not chosen to take a back seat to escape early, because I think it will be interesting. There are more than 7,000 PubMed-listed publications in the field of primary sclerosing cholangitis. So this is really impressive. And if we try to drill it down to one take-home message, it is this: there's no cure or specific treatment for PSC. And this has been published by the American Association for the Study of Liver Disease 10 years ago. But I don't think that in the meantime, there have been major breakthroughs which would challenge or change this concept. Therefore, patients with primary sclerosing cholangitis do not have choices. It's a rare disease. After 10 to 15 years, they're either dead or have required liver transplantation, and they have an excessively increased risk to develop cancers of the liver, the bile ducts, and the colon. Therefore, it's a really bad disease. And I'm not a hepatologist, but not becoming a hepatologist could be driven by this disease, because it's really frustrating to deal with this. Therefore, these patients so far have no new choices. And what you see here is what actually changed my mind. And this is a picture of a real colonoscopy. The name has been wiped out, but the date was the 1st June 2017. And at that day, this was a Thursday, because on Thursday I have my endoscopy list. And Dr. Shah, who is here in the room, was at that time the fellow in the department. And she called me into the room that I should have a look. And she said, look at this colon. This is a patient with ulcerative colitis. And I said, BS, this cannot be ulcerative colitis, because you rarely see such a complete remission with the normal treatments. Therefore, this looks like a normal colon. And this would be a picture of the same patient earlier, very intense inflammation, lesions. Therefore, everybody, even if you're not a gastroenterologist, you will appreciate this. This looks completely different. And I could show you now what are the effects of established therapies for ulcerative colitis, immunomodulators, but they're working. They have a significant gain over placebo, but you need to be really lucky to get such a response. And this patient didn't only have some response with regard to his colonic inflammation, there was also response to antimicrobial therapy, which was given for other reasons. And what you can see here, how the different liver function tests improved. And this was mainly driven by my colleague here, who much earlier than us has recognized this and started very systematically treat patients, children with ulcerative colitis and PSC with antimicrobial therapy. Therefore, if anybody is to blame that we are here tonight, it's him, not me. Therefore, he was a person who has initially really driven this. And therefore, here you see antimicrobial therapy has a very nice effect. But when we looked at the literature, there was more about this. Many years back, actually 1959, before I was born, there was this study from Sydney who observed obviously antibiotic therapy in patients who had a combination of ulcerative colitis, also had inflammatory changes of the liver, and amazingly, these patients responded to tetracycline. All this triggered our interest to do a systematic review of antimicrobial therapy in these patients, and not very good studies, no prospective randomized trials, but a lot of cohort studies. And these are the data. What you can see here, antimicrobial therapy works, and vancomycin seems to be the best drug. And subsequently, we developed a framework around this, how this could work. We published this in the American Journal of Gastroenterology. And this is a summary, and hopefully we can discuss this a little bit later, that there's a combination of genetic susceptibility, that there is changes of the biliome, changes of the microbiome if you treat patients with antibiotics. And in the end, the pathomechanism is that you have an inflammatory process with abnormal trafficking of lymphocytes from the gut to the liver. And the task which we see now is to find in the needle in the haystack of the microbe or microbes which are relevant for the progression and manifestation of this inflammatory process. What will help us is this Queensland-wide prospective randomized NHMRC MRFF funded study where essentially all major gastroenterology centers across the state participate. And I hope in the not too distant future, we will report more insights, not only about the response predictors, but also about the microbial and the immunological factors. And without any further ado, I would like to introduce Peter. Now the next one is Peter. Baby steps towards understanding pediatric PSC. And as I mentioned, Peter is to be blamed that we're here, and he has a long-standing interest in this, and he has done really landmark study and work from a clinical perspective to prepare the field. Peter.
Thank you. Well, that's a very generous introduction, Gerald. Thank you. Look, I've got 10 minutes to try and give you a bit of pediatric. Here's my thing. Bit of a pediatric feel for this space, and give you a little bit of introduction of how we've arrived at this point. And I can flick from here, can't I? So my talk is towards treatment for primary sclerosing cholangitis, baby steps towards understanding pediatric PSC. Let's go forward. Click this one. Cool. Just click on this one to make it. Okay, this is what we're talking about. PSC, primary sclerosing cholangitis, an inflammatory condition of the bile ducts leading to scarring, stricturing, inflammation, liver damage, in some cases going to cirrhosis, end-stage liver disease, and liver transplant. In some cases, malignancies of the biliary tree. What we're going to talk about a little bit as well is the associated phenotypic colitis that often goes with that, and I'll be telling you a little bit more about that. Predominantly right-sided colitis, often with backwash ileitis in continuity with an aggressive colitis, and characteristically rectal sparing. So PSC and UC. Sorry, got to get the right one. Where have I taken that off again? Do you want to give me the, just keep the thing. All righty. So I'm going to speak more as an IBD specialist than a liver specialist today with an IBD perspective, although it's of relevance to all of us in the room who deal with either liver or inflammatory bowel disease conditions. In the IBD clinic, the IBDologist will expect that 6 to 10% of their patients will develop PSC, and the hepatologist in his liver clinic or her liver clinic seeing patients with PSC will expect 75% of patients to have or develop colitis. So it's quite confusing from phenotypic entities and studies of prevalence if they don't always start at the same time, and that has caused quite an issue. What I will talk to you about is a little bit of the increased mortality and morbidity of having PSC with UC, some of the natural history in PSC in children, which is bad. To try and persuade you that PSC is not an extraintestinal manifestation or association with CE. But in fact, that conduit, that combination of PSC and UC is a distinct entity. And there is a typical but not pathognomonic phenotype of colitis in patients who have PSC. And that'll lead us on to the last couple of minutes of new drug, new therapy with an old drug, or of vancomycin in our experience. I'm just going to briefly show you a couple of slides from this epidemiological study to highlight the importance of PSC. It's a natural history of pediatric onset inflammatory bowel disease. Two key slides. The first one is that when you look at death in the following 20 years of follow-up after pediatric onset inflammatory bowel disease, the biggest cause of death by a long way are disorders of the GI system. So GI cancers, liver transplant, far, far in excess of the complications of therapy, the immunosuppression, the skin troubles, lymphoma, a virtually 50 times increased hazard ratio if you of death and troubles from your GI tract. And the other slide I want you to take away is the biggest risk factor for death in the next 20 years, more than your era of diagnosis, more than your age of diagnosis, whether you have surgery, whether you have perianal fistulizing disease, is whether or not you have PSC with your IBD, which gives you over 12 times standardized mortality ratio. And that information was reinforced with the European Registry of Deaths and Cancers in Children earlier on the last decade, showing that PSC was overrepresented with the fatalities in that 20-year follow-up. There are only 27 deaths, so deaths is not a common thing in young children. One of the commonest reasons for death is accidental, so all causes 27. When it came to fatal cancers, PSC is well overrepresented. All three colorectal cancers had PSC, all three cholangiocarcinomas had PSC, one of the patients died from liver failure as a result of PSC, and one of the 3 suicides had PSC. All righty, so let's talk a little bit about the natural history in children, which is quite similar to the natural history in adults, but quite catastrophic. This is a Canadian study from only a few years ago of nearly 800 children over nearly 4,500 patient years, median onset 12 years of age, slight male predominance, so strong association with 75% with IBD and mainly large duct disease. But the take-home message is that within 10 years of diagnosis, over 40% had had a major event, major being a pulmonary hypertensive complication, bleeding varices or ascites, a biliary stricture requiring dilatation, cholangiocarcinoma in 8 or 1% of those patients, liver transplant in about 1 in 6, and death within the first 10 years of follow-up in 11 out of those 700 patients. So quite a significant impact. I'm not going to talk too much about pathogenesis. We do know there's a little bit of genetic predisposition. We're going to hear a bit more about the immunological responses, and we're going to hear a bit more from Mark about the idea of a disturbed gut microbiota. But I'm going to show you a couple of slides which are quite interesting. One is a genetic slide, a very simple genetic slide, which tells us that while the genetic risk factors for PSC are not particularly strong, their genetic profile is distinctly different from those in the inflammatory bowel disease clinic. The other ones, patients who have UC or Crohn's disease, but not with PSC. Very different. You might have thought it would overlap a little bit with UC, but it has very little to do with generalized non-PSC UC or generalized non-PSC Crohn's disease. It has more to do with PSC. And again, when we look at the fecal microbiota, comparing those with ulcerative colitis alone, as far as can be determined, those with known PSC and those with PSC and ulcerative colitis, their microbiota signatures look far more like the patients with PSC than the patients with inflammatory bowel disease. So it's interesting that this isn't just an extraintestinal manifestation of inflammatory bowel disease. This is probably a very distinct entity. So when we look at the more typical or characteristic phenotype of patients with colitis who happen to have PSC at the same time. This is a study from Canada, a multi-center study, reasonable numbers, just under 100 patients. And some of the things that were coming through are things that you've obviously seen yourself in the clinic. But a common feature is that patients with PSC-UC tend to report being in clinical remission. They don't tend to have an aggressive type early on in particular. So 73% reporting they're in clinical remission at any one time. At the first colonoscopy, there's a much greater type of rectal sparing. So classically in inflammatory bowel disease, especially ulcerative colitis, you expect the worst colitis to be in the rectum, progressively milder as you go more proximally. Typically in this group, the rectum is often spared. and they get more aggressive right-sided colitis with extension into the ileum with backwash ileitis. And so there's a much greater incidence of a worse colitis proximal to the rectosigmoid. If we go back to the ones reporting that they're clinically well, if you do scopes across the board at a cross-sectional moment, they have a much greater rate of actual endoscopic disease. So milder symptoms, milder reported patient-reported outcomes, far more commonly actually having active disease in over 80%. All right, talking a little bit briefly before we go on to vancomycin about the true prevalence of PSC. Systematic reviews reported as between 6 and 10% of the inflammatory bowel disease clinic. In our UC population, it's virtually 20%. That's because we have a very high rate of MRCP performance when we see patients with persistent abnormal LFTs, or this increasingly apparent PSC colitis phenotype that we're starting to use that terminology. And an interesting study informs us how difficult it can be to work out the prevalence, comes from Norway. This was published in Gastroenterology a few years back. A large Norwegian clinic where they had over 300 adults who'd been followed up for 20 years. They all had routine MRCP and colonoscopy. And before their MRCP done just across the board, they reported their prevalence of PSC as 2%, 7 out of those 322 adults. After they'd finished doing their MRCPs, their prevalence went up to 8%, 4 times increase, with 17 new patients diagnosed with PSC. 65% had had normal LFTs throughout their entire follow-up. So the more you look, the more you find. That confounds our understanding of the epidemiology and prevalence and associations of this condition. So PSC-UC is a wolf in sheep's clothing. It has a gentler clinical presentations. A lot of patients with active colitis really reporting clinical remission, and a lot of patients with active PSC having normal LFTs. Yet you can see there's a lot greater adverse outcomes for both the colon and the liver, and we haven't gone on to colectomy and colon cancer, but that's certainly the case. All right, as Gerald has told us, there are none established treatments for PSC. There have been multiple small studies and reviews about various antibiotics for the management of PSC. Even ursodeoxycholic acid has come in and out of fashion. And oral vancomycin seems to be the one that's had the most promise. But the problem with oral vancomycin, any therapy, there have been very unclear liver endpoints. A recent pediatric study using the somewhat controversial technique of propensity studies concluded that there was no difference in patients treated with ursodeoxycholic acid or oral vancomycin or no therapy over 4-year follow-up. 2 issues with that. Propensity is where you take a whole group of patients who are matched for their severity and phenotype of their disease, who happen to be treated differently because of different consultant or centre practice, and see how they all go. There are definitely flaws in that, but the biggest flaw in this study is that 4 years probably isn't long enough to work out the difference in those outcomes. 10 years is catastrophic, but the first few years you're not probably going to get enough differences to give you a good differentiation. So that's the backdrop. But now we come to the last couple of minutes, where we are now and how we got there. Because our interest in PSC came about principally because of the colitis. And it starts with a couple of very small reports among those thousands that are reported in the pediatric literature about the use of oral vancomycin in a couple of children with PSC. When I say very small reports, this was the same group, but over 14 years, they reported on a total of 14 patients. So hardly definitive or landmark studies. They reported improvements in the LFTs over the years. But what caught our attention was the dramatic improvement in the colitis in those patients. And that became very important for this particular patient who's really heralded our use of vancomycin in this setting some years ago. And I'll take you briefly through her story because it is quite striking. At 18 months of age, she presented with autoimmune hepatitis. Her biopsy and her imaging suggested she had an overlap. She had autoimmune hepatitis with interface hepatitis and plasma cells, and she had PSC with inflammation of the bile ducts, a little bit of onion skinning, and that autoimmune hepatitis proved particularly medically refractory. At 7 years of age, she had her first transplant, which all went very well. In the explant, it showed that the histopathology was all PSC. There was no autoimmune hepatitis, which is actually quite a typical story for PSC in pediatric practice. Increasingly, we're realizing that it's mostly about the PSC. There's a little bit of overlap inflammation, and see some plasma cells, but almost always it goes towards PSC, not autoimmune hepatitis in subsequent follow-up. Anyway, 2 years after a transplant, she had some occasional blood in her stool. Colonoscopy showed a mild right-sided colitis, which got even better before we talked to the patient about some therapy. We recommended they think about 5-ASA. They didn't use it because all the blood went away. However, 5 years later, 3 years after the blood, but 5 years after her first transplant, she required a second transplant for a recurrent PSC in the graft. Again, 2 years after that, she had an aggressive inflammation in her liver. She had a biopsy which showed aggressive fibrosis. She started developing some portal hypertension. She had some occasional blood in her PI. She had another colonoscopy, again, which showed a moderate right-sided colitis. There was no response to standard therapy. She was already on immunosuppression, being a transplant candidate. And the surgeons, not unreasonably, suggested it would have been standard practice, and is in many cases, you need your colon out. If your liver's going to survive or have any chance of surviving this liver or your subsequent liver, you need to lose your colon. So a colectomy for a current PSC is well recognized. And her response was literally and metaphorically, over my dead body. So we then looking around for other alternatives. How can we treat her? We came across these two papers. We gave her some oral vancomycin. The colon completely returned to normal, complete mucosal and histological healing. Her LFTs improved. She was then followed up for another 4 years in good shape, but I suspect we were just delaying the inevitable because sometime after transition, she had her third transplant. But nevertheless, the mere fact that we could control her colitis completely with an agent such as vancomycin led to a lot of enthusiasm, and many of you will have seen this data before. It was published in Gut. I've got some follow-up data to give you in the last couple of minutes. But basically, we started to throw it around a little bit. We had patients with PSC phenotype and confirmed PSC that were having failed conventional therapy. We had another young lady who had recurrent PSC in her transplanted graft with a right-sided colitis. We had a couple of patients who deferred conventional therapy for vaccination. We had 3 patients who had primary therapy because of overzealous junior doctors. But the outcomes are quite striking because in 17 patients, this was every patient we used oral vancomycin for, except for one that only had 2 weeks before their colectomy. 17 out of 17 had complete clinical remission. Doesn't always mean that much in this setting, but 15 out of 15 who had paired calprotectins returned to normal, less than 100. 15 out of 15 who had paired colonoscopies returned completely to normal. 9 out of those 15 microscopically normal too. The LFTs, there was some striking improvements in a number of patients, but by no means as complete. So only 12 out of 17 improved, particularly the 2 that had their liver transplants, one of whom you heard her story. So, ironically, as at the same time I'm presenting to yourself, my colleague Dr. Belush is presenting to the World Congress on Pediatrics this same data, and she presented it last week to DDW, our 2-year colitis outcomes with oral vancomycin. Of course, we got 5- and 6-year outcomes, but we've only got decent numbers that we can reliably talk about for 2 years. These are all our patients, and all that received at least 2 years of therapy. Children under 18 years confirmed PSC with liver biopsy or cholangiography or both, and ulcerative colitis with active disease, calprotectins, and in many cases colonoscopy, given oral vancomycin for their active symptoms. They were given, only children receiving a minimum of 3 months were reviewed. The main indication now was failed conventional medical therapy versus the sporadic ones that we had before of primary therapies and what have you. We defined deep remission as steroid-free, complete abolition of symptoms, and a normal calprotectin less than 100, and/or most patients had calprotectins, and about half had repeat paired colonoscopies. So very strict endpoints. And out of 29 children, after 6 months, 27 were in deep remission, 2 didn't have a response. 12 months later, the 25 of those 25 in deep remission that responded, 25 were still in deep remission, 5 of them were retreated, 2 were lost to follow-up. At 2 years of follow-up, of those 25 children, we had data on of those 25, we had data on 22. 15 were still in deep remission, normal calprotectins, normal symptoms, and those that have been retreated have to be retreated. 5 had lost response or were retreated but didn't regain their response. There was no data on 2. I wish I could tell you more data about their LFTs and imaging. There's a lot of data on their LFTs and imaging, a mixed bag of striking results and no results, which takes a bit more analysing, so I can't share those with you yet. No patient came to colectomy. Remember, these are all patients failing conventional medical therapy, and no patients developed VRE. So in summary, hopefully I've been able to persuade you, and hope not gone too much over time, a bit, that PSC is a catastrophic diagnosis in the IBD clinic, and not such a great diagnosis in the liver clinic either. It's the main association for death and cancer in paediatric onset IBD. Its relationship is underreported as in the early stages. It's both aspects are clinically mild early on. But I think we're increasingly seeing that this is a distinct entity from other forms of inflammatory bowel disease, as it has different genetics, microbiota, and an emerging phenotype, and therefore warrants different treatment strategies such as antibiotics. And hopefully I've shown you that oral vancomycin is a very useful therapy. Thank you.
And without any further ado, I would like to invite and introduce Mark Morrison. He is the chair of Microbial Biology here at CTI and certainly a world leader in his field. And his presentation is entitled A Micro Look at PSC. Mark, please.
That's it. Yeah. All right. Okay. So good evening, everyone, and nice to talk to you about a micro look at PSC. And what I'll do with the 10 minutes that I hope I stick to is give you an overview of what might be the current state of knowledge and the technologies that have been used to achieve that, and what we're trying to do here at joint efforts with PA Hospital and TRI in terms of taking a step forward in this space as it relates to some of the observations and findings that Peter has talked about in the clinical setting. So my disclosures, I just want to point out, I have PhD training in microbiology and no medical qualifications. So just excuse the words and language I use from time to time. And I've just been asked to give a talk on dysbiosis and IBD for a conference overseas. And I'd done a scope of search for this. And it found more than 800 documents using those two words as identifying the topic and the search field. And you can see the exponential growth of the field that's taken place since 2009, which is really driven by the application of culture-independent DNA sequencing technologies. So this afternoon, I thought I'd take a look at dysbiosis and PSC as search terms. And I found a total of 20 documents published over the last 5 years. Now, if I'd used the word microbiome and PSC, I would have clearly increased the number of publications, but only by a factor of about 3 or 4. So the focus on this particular disease or clinical entity and looking at the microbiome is both relatively recent and not well examined or studied at this point in time. This is from our review article to kind of stimulate the field in terms of thinking about PSC a little bit differently and more holistically. And it takes advantage of a number of studies that have been published over those last 5 years that take a look at microbiota profiling, either of stool samples or colonic tissue samples, and the changes that they've seen in the microbiota of those individuals compared to healthy control subjects. Now, these data are useful and relevant, but the current state of the art of this technology means that you really only have a description of the type of microorganism based on its taxonomy. In other words, you have a name of a microorganism, but you don't have any kind of functional information in terms of how the capability of that microbial culture or community is changing. And the second thing associated with this particular work is that you still don't have a sense in terms of how much microbiota load is associated with these particular samples. And indeed, is that relevant and important? And I think in the next couple of slides, I'll try and demonstrate to you why it is and how it can make this field of microbiome research advance in more definitive ways in terms of addressing a particular disease entity such as PSC. So microbial diversity and load on gut tissue varies. And so I've shown you some various micrograph shots here of tissue taken from the duodenum on the left-hand side and at various segments along the intestinal tract and into the large intestine as well. And you can see just simply by the shape of the microorganisms that are present on that tissue, as well as the numbers of them, that there is variation, not just in microbial diversity, but the load that is present on this tissue. And I think that we're going to have to start examining microbial load associated with these communities just as much as we do the diversity of the populations or the communities. And so how are we doing that here in Brisbane? This angry looking man in the photo you've seen is the chair of our session tonight. And he invented this device, which is now patented and that we call the Brisbane aseptic biopsy device. And what the characteristic or unique feature of this device is that the forceps reside within a sheath, which is plugged with a glycerol plug. And as a result, keeps the forceps inside a sheath that's sterile until it physically reaches the side of the tissue where you want to collect the sample. At that point, the forceps are extended, the tissue sample is taken, and the forceps are retracted, loaded with the biopsy tissue back inside the sheath. And basically what we feel and have shown through a number of publications is that this collects the tissue in what I call an aseptic way. In other words, the tissue itself has the microbes that are physically associated with the tissue, but it spares that tissue from potential exposure to either luminal contents or other contaminating factors during the endoscopy process. And as a result, two things can happen. We can start to use a broader variety of techniques to examine the microbiota associated with that tissue, with the confidence that it's not contaminated with other microorganisms we're not interested in. The other thing it does, especially with the hospital so close to TRI, is that we can either preserve the samples under a sterile anaerobic condition for long-term storage, or we can bring the entire device back over in a plastic bag filled with carbon dioxide and immediately initiate the cultivation of the microorganisms that are physically attached to that biopsy tissue. So this device expands the scope of work that we can do, not just taking or extracting DNA from that biopsy and using PCR-based approaches to give you something of a stamp collection of what types and kinds of microbes are there, but also some sort of quantitative measure of how many microbes are physically present on that tissue, and also start to recover that community in a culture-dependent way that gives us a holistic representation of that community as live microorganisms, which can then be used in various in vitro or other sorts of studies to examine the functional attributes that that community provides at the site of the tissue that's been sampled and the clinical phenotype that might be associated when that tissue is collected. So just a couple of examples. These are some unpublished data produced by work driven by Aisha Shah. And what we've looked at here is using 16S profiling with tissue collected by the aseptic device. We've been able to identify that, as you would expect in healthy control patients, there is an increasing amount of microbial diversity or bacterial diversity on tissue collected at different segments as you move from the beginning to the end of the intestinal tract. And so that's what we would expect. So it's a good kind of control in terms of what the community diversity should represent. But you can see in the 16 subjects where we've used this aseptic device and collected the tissue samples, we find that the diversity scores are generally lower at each individual segment compared to healthy controls. And in particular for the duodenum, we approach statistical significance in terms of that change in diversity, but we definitely do at the right colon. So again, the point I'm trying to make is that by using this particular device and using some of the standard sorts of techniques and measurements, we get a representation of the microbial community in terms of how it's changing along the gastrointestinal tract associated with this disease entity. And in addition to these changes that we see within the large intestine or the colon, there also appears to be meaningful changes happening in the duodenum as well, on the tissue. So as I said before, the other advance that this sort of device provides is the capacity to store a tissue sample under anaerobic sterile conditions for subsequent cultivation of the microorganisms. And that's what I'm going to talk about briefly just now. And so in addition to the capacity to culture the microorganisms, we can extract the DNA from that culture and compare it to biopsies that have been preserved and the DNA extracted directly from those. And as a result, have a microbial enriched data set that represents the functional attributes of the entire community that we've been able to recover from that tissue. And as a result, start to understand the functional context of the dysbiosis that we appear to see associated with PSC and in comparison to healthy control subjects. So just very quickly, we've published the actual methodology of this in the last week or two in the ISME journal, but looking at biopsy tissue collected by the PA group, you can see in the left-hand side our microbiota profile using the culture independent based approaches, and then the microbial culture metagenomic sequencing approach that we've applied to the match biopsy sample stored in glycerol indicates that we have a very good and holistic representation of the biodiversity from tissue, and we can culture it again and again and again and retain that sort of compositional framework in a manner that allows us to do repeatable studies in vitro with live microorganisms, as opposed to just using DNA sequencing data. The other advent or opportunity that this technique's providing us is to look at what I said is bacterial load on tissue. And again, as part of Aisha's PhD program, she's collected tissue biopsies using this device. When we extract the total DNA from that biopsy, we can apply quantitative PCR that targets not just the beta-actin gene and the number of copies in the DNA sample, but also a primer set that recognizes the bacterial world specifically and provides us a copy number of that amount of bacterial DNA within the total DNA extracted from the biopsy. And by proportional representation to beta-actin, we can kind of normalize the microbial load score on that tissue. And the kind of so what, who cares about this is that what we found in a larger study of a variety of cohorts, either suffering from a functional gastrointestinal disorder or Crohn's disease and ulcerative colitis, compared to those that have gone through endoscopy for surveillance or precautionary procedures that proved negative upon those examinations, we see that those with a functional GI disorder have a lot more bacterial load on tissue at the duodenum compared to the so-called control group. And interestingly enough, even at the duodenum, we see a much reduced load of bacterial biomass, both in the Crohn's disease and ulcerative colitis patients. And we haven't published the additional work at this stage, but when we look at samples taken in the same way and analyzed in a similar manner from the terminal ileum of these same cohort of subjects, we see similar trends. So there's evidence that on mucosal tissue, at least, we need to be thinking as much about how much bacterial burden is present on that tissue and how that amount of bacterial load is changing or responding in response to host signaling as well as environmental changes within the GI tract. So I want to take that to a final step and talk about some work done by Jeroen Reis at KU Leuven, who's a collaborator on our CRE in digestive health, supported by the NHMRC. And in addition to the work we've done over the last 4 or 5 years to look at bacterial load measurements on mucosal tissue, they've developed what's called a quantitative microbiome profiling technique to use with stool samples. And so very simply, the stool sample, the DNA is extracted and used by the various methods of DNA sequencing technology. But before doing those extractions, a matched subsample of that stool is diluted and filtered to remove large particulate material. And then by using flow cytometry, they get a count of the total number of bacterial cells that may be present in that sample. And what I'm trying to show you here on the left-hand side is pretty clear evidence with statistical significance that compared to healthy controls, which is the left-hand bar graph, persons with PSC alone have a reduced microbial load in stool samples, but those with either UC or Crohn's disease in particular have further reductions in microbial load in stool, suggesting that environmental changes are taking place, probably as much to do with transit time and the movement of digester through the intestinal tract as anything else, that influences the number of microbes that are actually physically present in stool. And it's important to take these sorts of quantitative measures into account to move beyond just looking at the compositional framework of the community, which looks at relative abundance. As you might imagine, you may have a percentage increase from 1 to 10% in a specific bacterium using just the compositional profiling techniques. But if microbial load has increased by tenfold, the numbers of the microorganisms present there are effectively the same, okay? So it's important to start to think more carefully about population size and a quantification of that as much as the composition and the profile. And that's demonstrated by the figure here on the right. So basically they've looked at various measures here. They've looked at fecal moisture and they've looked at biomarkers for IBD, calprotectin, and they've looked at biomarkers for liver function in the form of ALP. And as you can see, as you move from left to right, these are the most predominant and discriminatory bacterial taxa that they're able to identify in their cohorts. But what they found is that the bulk of the effect size associated with the IBD phenotype was associated with these 4 bacteria towards the end here. Fusobacterium, Escherichia, Streptococcus, and Bartonella, whereas the changes in liver function are linked most predominantly with Enterococcus. Why is that important? Well, Enterococcus is the major target of an antibiotic like vancomycin. So here is some evidence indirectly where you can connect some dots between vancomycin utilization, targeting Enterococcus populations, and having an impact on liver function as opposed to the actual IBD symptomology that can co-occur with this condition. So I just want to say thank you. I think I'm done, Gerald. Hopefully I'm close to 10 minutes. Just recognise those collaborators I have across Australia in various areas, but in particular those at PA Hospital, and thanks for your attention.
Thank you, Mark. Brisbane is, I would say, a hepatology hotspot. And if you look at the map worldwide, Brisbane stands out with the hepatology research. Beyond doubt, one of the thought leaders, the intellectual thought leaders, is Gerald Crawford. And Gerald is sitting somewhere back. And one of his long-standing partners in crime is Kim Bridle. And Kim is here today to speak about PSC, a PSC model. Kim, thank you.
Thank you. I'm going to start by admitting that I'm neither an immunologist or a clinician, so I'm not sure why I'm speaking at an immunology in the clinic forum, but nevertheless, I'm going to give you a quick introduction into models of PSC, in particular the MDR2 knockout mouse. Can everyone hear me? Yeah. So there's been various models of PSC developed over the years. There's been chemically induced cholangitis. There's been the knockout models, of which the MDR2 I'm going to talk about today. There's been the use of infectious agents, There's been experimental biliary obstruction models, so the bile duct ligation model, which I'll also touch on, which is, I guess, the most commonly used model of biliary injury. Not that it's a very good model of PSC specifically, though. There's models involving bacterial cell wall components and colitis. There's endothelial and epithelial cell injury models and antigen-driven models of biliary injury. None of these are perfect. So there's a lot of limitations to these various models. There's high mortality, poor tolerability. There's no large duct involvement with a lot of these models. Intestinal obstruction, they're either very complicated or technically challenging, or the disease phenotype is very mild. So no fibrosis or low levels of inflammation. And in the case of the MDR2, no development of cholangiocarcinoma or IBD. And so the model, as I said, we've got the most experience in is this multi-drug resistant to knockout mouse model. And so how does the injury develop in this model? So in the MDR mouse, they secrete phospholipid, they don't secrete phospholipids. And so the bile acids can't form these mixed micelles with the phospholipids. And so the toxic bile acids are just roaming around free. And so those bile acids damage the tight junctions between the biliary epithelial cells, and they also damage the where am I going? Damage the basement membrane. So the bile acids leak out into the periductal area. This recruits inflammatory cells, neutrophils, macrophages, and lymphocytes, and you get this activation of these portal myofibroblasts around the ducts. And what happens there is it completely surrounds the duct tissue. So you get this classic onion skin fibrosis around the ductule. And what that does is really separates the biliary epithelial cells from the blood vessels that supply those cells. And so those cells eventually die. And that causes this fibro-obliterative injury. And so the use of MDRs in research really are predominantly as an antifibrotic model. And so lots of people have looked at this model, to look at antifibrotic therapies of various sorts and to study the evolution of fibrosis. And this is mainly where we've done our research. But more recently, I guess, there's studies using this model to look at molecular mechanisms of HCC development and to look at inflammatory hepatocarcinogenesis. And even more recently, there's been a couple of papers on gut dysbiosis and in the development of HCC and in the inflammasome activation and injury development. And so I think that's probably 2 or 3 of those, 20 papers Mark was talking about maybe. And so our work has really looked at the evolution of fibrosis. So the fibrosis develops quite early in this model. So at about 3 weeks of age, you get this sort of mild fibrosis around the portal tract. But by 16 to sort of 8 to 16 weeks, you get advanced fibrosis and early cirrhosis in this model. But then it sort of just reaches a peak and doesn't progress further. And so when you look at either hepatic hydroxyproline or fibrosis stage, by about 8 weeks, it reaches a peak and then either plateaus off or even decreases slightly. Likewise, the ductular reaction, which peaks at about the 5 to 8 weeks, but again, plateaus or decreases. Inflammation, again, peaks at 8 weeks and then decreases. It never goes back to normal. And so you've got this chronic low-level inflammation going on in these animals. What we found is when you look for gene expression of pro-fibrogenic mediators, they really peak quite early and then drop off almost completely. But you do get, the fibrosis and the injury is maintained with time. And even later still, you get some sort of matrix remodeling happening with these animals. And so the MDR2 model is a good model of progressive fibrosis. It's reliable and reproducible and probably more closely replicates that the fibrosis development seen in humans compared to other models, particularly if you compare it to something like the bile duct ligation model. But we've previously used both models to look at antifibrotic agents. And so when we looked in a BDL model, we treated these animals with rapamycin, it's an immunosuppressive agent, rapamycin. We found significant reductions in fibrosis score, significant reductions in hepatic procollagen expression, significant reductions in the ductular reaction. But when we repeated these experiments in the MDR2 mice, nothing. So we're just looking at these two bars here. So when you look at hepatic hydroxyproline, treatment with rapamycin did not do anything. There was no change in fibrosis score, and there was no change in the ductular reaction. And so we're not really sure what that is. It's whether, you know, there's so many factors playing a role here, whether it was the pathway we were targeting, whether it was how we did this experiment. We tried treating rapamycin at the beginning of the injury process and later in the injury process, but it didn't make a difference. So I guess it's just a cautionary tale about not all animal models that you think are doing the same thing are. I'm not going to touch very much on cancer development and MDR2s, but just to point out that there's no cholangiocarcinoma development in this model, but HCC does develop from about 9 months, with about 100% of the mice being affected by 12 to 16 months of age. So you can see here multiple nodules within this liver in I think that's a 15-month-old mouse. And so inflammatory responses in the MDR model have been examined in the progression to HCC. This paper from a Sydney group showed that as injury develops, so they've looked at the gut microbiome and liver inflammation and its role in HCC development. And so they've just shown as injury develops in these MDR models, there's a real change to a Th1, Th17 pro-inflammatory cytokine profile as disease progresses. And this same group demonstrated that while there's a real shift then from a pro-inflammatory phenotype to an immunosuppressed profile as HCC develops. This paper also looked at a lot of sort of gut profile as well. So. And so again, there's not very many studies solely focused on the immunophenotype in MDR2s. This study in hepatology a few years ago showed that the Tregs, so the red line here is the MDRs and the blue line is the control animals. And that with time and disease progression, there's a real contraction of the intrahepatic Tregs in the MDR2 mice. And they've shown that the hepatic CD8 cells drive this biliary injury. And so when they used a CD25 Treg depleting antibody, there was an increase in the CD8 cells, which also increased the ductular reaction and increased injury in this model. And they found that interleukin-2 could reverse that injury progression, decrease ductular proliferation and decrease fibrosis. And so this mouse model is a good model of inflammation-mediated hepatic carcinogenesis, and they've shown that dysbiosis and altered function of the microbiome occur as injury progresses. As I mentioned, there's that shift to a Th1, Th17 phenotype, and there's an immunosuppression as HCC develops. Now, I've stolen this same image that Peter showed and just saying to finally say, does the pathology of MDR2 replicate PSC? And while they are a good histological model of PSC, they do lack cholangiocarcinoma and IBD. And this is a very complex disease and with a lot at play, as Peter said, genetic factors, environmental factors, this inflammation, the fibrosis, and we can't forget the bile acids. And so I think it's one of the best models we have of this disease, but it's not perfect. Thank you.
Thank you very much, Kim. This was very interesting and really bringing the basic science aspect to it. And now I have the pleasure to introduce the last speaker, Kate Lynch from the Royal Adelaide Hospital. And I know Kate for many years when she applied for her training position as an advanced trainee in Adelaide. At that time, probably the vision to do a PhD and to do research was quite remote. But when I had a chat earlier today with her, I was very pleased to hear that she's infected with the research bug and that she is really excited to present today. Therefore, Kate, thank you very much for being available, and we all look forward to your presentation from the gut to the liver, T cells in PSC-IBD.
Thank you, Gerald. I'll just share my screen. Right, hopefully you can see that. So I'm delighted to be talking to you today about my PhD work of T cells in PSC-IBD. And here are my disclosures. So I'll be talking about gut homing markers and their role potentially in PSC. I'll introduce you to my hypotheses I had at the start of my PhD, and then my main results in a very kind of flagship view of very quick whistle-stop tour of my PhD of what I found in peripheral blood, gut and the liver. And I'll also touch on the effect of vedolizumab in PSC and future directions. So when we're talking about gut homing molecules, we're talking about CCR9 and alpha-4 beta-7 in particular. So CCR9 is a chemokine receptor on T cells, and when it connects with its ligand CCL25, which is secreted by the intestinal endothelium, it causes the lymphocyte to slow down within the blood vessel within the intestine and go more slowly. And it also causes expression of an activation of alpha-4 beta-7, which then binds to its ligand, MAdCAM1, which causes it to arrest in its movement and be extravasated through into the lamina propria of the intestine. And you can see here the schematic with these 4 key proteins. Now, 20 years ago in Birmingham, Grant and David Adams' group showed that MAdCAM1 was expressed in intestinal endothelium, that it wasn't present in the healthy liver, and that MAdCAM1 is the culprit for attracting alpha-4 beta-7 cells to the intestine, like I just showed you. And Grant then went on to demonstrate that there were alpha-4 beta-7 positive cells in the diseased liver using explants. And he also showed the presence of MAdCAM1, and actually the highest presence of MAdCAM1 he found in PSC compared with other liver diseases. And I've shown you some of the relevant immunohistochemistry here. Furthermore, a few years later, Bertus Eckstein in the same lab with David Adams published his work from his PhD, where he took explants from patients with PSC and with other liver diseases, and also looked in peripheral blood and in the intestine. And when he analysed PSC liver infiltrating lymphocytes, he found that 20% of them were positive for this gut homing marker CCR9, compared with only 0 to 1% of normal liver infiltrating lymphocytes. And as a positive control, many Crohn's disease intestinal lamina propria lymphocytes. So this led me to nothing much was done since then. And so 10 years later, I developed these hypotheses that there is a differing distribution throughout tissues, peripheral blood, intestine, and liver of gut homing lymphocytes in PSC-IBD versus controls, that these lymphocytes accumulate within the liver in PSC-IBD, but not in control livers, that vedolizumab, which targets these gut homing cells, can cause a reduction in alkaline phosphatase, a clinical marker, in a subset of patients with PSC-IBD, and that vedolizumab alters the distribution of these gut homing lymphocytes in IBD by reducing their prevalence within the bowel and potentially preventing their entry into the liver in PSC-IBD. So very briefly, I isolated T cells from peripheral blood, from biopsies taken at colonoscopy, and also from liver, predominantly from fine needle aspiration of the liver, a newer and less invasive technique than lower risk than liver biopsy. I performed flow cytometry and also immunohistochemistry, focusing on gut homing markers, and I compared patients with PSC-IBD versus various controls. And I also analysed some samples for patients pre and post vedolizumab. So we go to my peripheral blood data. You can see here that here I've got CD4 T cells within peripheral blood, and blue is the PSC, orange liver controls, ulcerative colitis patients, healthy controls, and these are small duct PSC. So I did find reduced CCR9 expression on CD4 T cells in patients with PSC compared with UC alone. And whilst there wasn't a difference for PSC for beta-7 as a surrogate for alpha-4 beta-7, when we look at T cells which express both CCR9 and beta-7, there was a reduction in PSC peripheral blood compared with UC in healthy controls. Furthermore, I found that there was a reduction in mate cells, mucosa-associated innate-like T cells, which are, again, gut-associated T cells in PSC compared with healthy controls in UC. So then if we go to the gut, where I extracted T cells from the lamina propria, first of all, to show you with flow cytometry that CCR9 positive CD4 T cells are much more prevalent in the ileum, moderately in the colon, and this is consistent with their gut homing capacity, and they're very low in peripheral blood. Now, this is a very bitty graph, but if I show you here, this is looking at CCR9 expression on CD4 T cells in the gut, in the ileum, the uninflamed colon, and the inflamed colon. And in blue is PSC, red is UC, and green is healthy. And so the only statistically significant finding, and very interesting finding, is that CD4 T cells in the ileum have reduced CCR9 expression compared with UC and healthy controls. And I wasn't able to find any differences in the colon or within beta-7 and mates in terms of expression. So if we go on to look at this with immunohistochemistry, CD3 staining with immunohistochemistry is very well established, but it's never been successfully done in humans in terms of CCR9 expression. But over time, I was able to develop an immunohistochemistry assay to positively identify lymphocytes which express CCR9. And I've given you some intestinal examples here of the colon and the ileum in various conditions, PSC, healthy control, and UC. You can see the clear CD3 expression and clear PSC expression on lymphocytes. And so I looked at the mean count of CCR9 and CD3 per high power field. And you can see there was no difference between PSC-UC, UC without PSC, and healthy controls in terms of CCR9 expression in the ileum and colon for PSC versus controls. However, and similarly, no difference with CD3 expression. However, if you go to look in the ileum and look at the proportion of CD3 cells, which is CCR9 positive, or the ratio, you can see numerically that there was a lower expression in the PSC-UC ileum compared with controls, although it didn't reach statistical significance. Now, immunohistochemistry is not the best tool to do quantitative analysis. I think the main thing here is that there's potentially a reduction in the ileum similar to flow cytometry, and that these cells are positioned in the lamina propria. I'll just go on to my observations in the liver. So these were derived from fine needle aspiration of the liver. And I looked at CCR9 expression on CD4 and CD8 cells in patients with PSC, as well as liver controls, which were people like NASH, PBC, and a variety of other liver conditions, alcoholic liver disease. And whilst I found no difference in the proportion of these T cells, which were CCR9 positive or beta-7 positive, you can see that actually in some groups there were quite relatively high numbers of CCR9 positive cells and beta-7 cells when these are not meant to be in the livers. So indicate, and actually a lot of these ones with high expression had high liver stiffness measurement, although unfortunately I wasn't therefore able to correlate this CCR9 expression with liver stiffness measurement. So in any case, I wasn't able to prove the theories that Grant and Eckstein put forward, and whether or not that is because I had this disease at an earlier stage of disease, whether or not it's a different technique. However, I think there are some clues here that all these livers are inflamed, and some patients have got high expression of beta-7 and CCR9. So now I'll just go to the immunohistochemistry. You can see I again stained for CD3 as well as CCR9 successfully. And we've got some PSC, PBC liver, PSC without IBD. And again, you can see that these cells are on the lymphocytes around the bile ducts in the PSC and are in the PBC. So pictorially, we can see where they are. Again, numerically, I found no difference in CCR9 expression and CD3 expression on immunohistochemistry between PSC with IBD, PSC without IBD, and liver control. So these are all on liver biopsies. However, if you start looking at the ratio again, the proportion of CD3 cells, which is CCR9 positive, you can see that it's actually higher in the PSC IBD patients, although not to statistical significance, compared with PSC patients without IBD, whether or not they're a different phenotype, and liver control. So just giving us some clues and food for thought. Very briefly, I did look with the International PSC Study Group at 102 patients retrospectively over a median of 400 days, patients with PSC IBD on vedolizumab, and looked what it did to their liver biochemistry. And you can see that over time, there was a rise in the mean ALP from equivalent of 240 to 306, which is not very promising, and similarly a rise in their transaminases, although a proportion of patients, around 20%, had a significant drop in their alkali and phosphatase. Maybe there's a subset of patients for whom it may be helpful. But I then went on and analysed my patients who, my liver samples that I'd done flow cytometry on, and looked at the memory liver T cells of the samples where patients were on vedolizumab versus not on vedolizumab. And whilst there was no difference in beta-7, there was raised CCR9 positive cells in the CD4 memory T cells for patients who are on vedolizumab versus not on vedolizumab. And some of these are biopsies, the majority are fine needle aspirates. So this was somewhat concerning about whether or not the vedolizumab was redirecting cells. So I'll leave you with a pictorial graphic of everything I've discussed, which is that in PSC, I found a reduction in gut-homing T cells in PSC IBD, and I've also found a reduction in the CCR9 positive gut-homing T cells in the ileum in PSC IBD. And whilst I didn't give you convincing data, there's a suggestion of a proportion of patients with raised CCR9 positive and raised, I didn't show you this, but raised MATE cells within the liver. So the hypothesis is that in PSC IBD, that the driver may still well be the gut-homing lymphocytes within the inflamed liver. In terms of future directions, I have done bulk RNA-seq of intestinal samples, as well as single-cell RNA-seq of liver samples. And I haven't shared with you those analyses today. Here's just a touch on some of the TSNEs, but I'm in the middle of analysing them and writing that up currently. And I think that a larger scale prospective study of vedolizumab is warranted, but not only looking at clinical outcomes, but experimental outcomes. So with that, I leave you, and I'd like to just acknowledge my supervisors and my collaborators. All of this work was done at Oxford and is ongoing, and thank you very much for your attention. Thanks very much.
Thank you very much, Kate. My task is now to have a group discussion and to wrap it up. I mean, time is advanced, but I still would like to take advantage of the presence of the experts here. And Kate, you will stay online for a moment?
Yes, I will, yes.
Wonderful. And may I ask the first question? And while everybody gets ready, Gerald, you are the hepatology intellectual leader in this area. Would you agree this is time for a paradigm shift how we approach PSC? Traditionally, it's considered a condition where you use immune modulators, lots of other things, but no breakthrough. Is this bringing the gut and the liver together? Does this change our approach?
Well, I think it's time for change. The PSC has been the black box of hepatology for 40 years, I think. And whilst there's been advances in many other diseases, particularly around viral hepatitis and PBC, we remain with the same problems with PSC patients that we had when we first engaged in the disease expression. So I just said that we've got the same problems with PSC now as what we did when we first engaged with the specialty. And so I think the time is right, the hypothesis that you put forward and that has been spoken about today seems very logical sequence. In fact, ties in the MDR model, I think, in terms of aberrant bile constituents contributing to disease. So I think that it's time to move the field forward, and I hope this is a bit advanced moving forward.
Thank you, Gerald. If I reflect of some of the things I've done in the past, many years back, I was involved in a study where we looked at predictors for relapse of peptic ulcer disease. And my task was to look at stressors. And when we analyzed the data, it was really striking that those who reported a cold, a common cold, which was then treated by the primary care physicians with an antibiotic, had a much reduced incidence of relapses of peptic ulcer disease. At that time point, Helicobacter was not known, and nobody could have dared to consider that peptic ulcer disease is triggered, caused by a bug. There's a possibility that one day we will have a single bacterium which interferes with the immune system, the bile metabolism of a toxic bile acid. Mark, what are your thoughts?
No, I don't know. Look, the precedent's been set, and I think even over evolutionary time, biology does the same thing kind of over and over again. So I think the likelihood of the chances are that it can happen. The linkage, I think, if we look at that data that came from the Lewindon group and the discrimination of the changes in the microbial community in a stool sample as a biomarker of particular taxa being associated with changes in liver function compared to changes that are linked more with IBD itself. And the role that vancomycin does appear to play does suggest that, you know, we need to think more carefully about that group of microorganisms in general and the relative abundance and the ecology of them throughout the gastrointestinal tract. And I really think that at this stage, stool is the end of the line. It's what you find. We need to be moving closer towards where the action really is relative to the clinical entity that we're looking at. And with PSC, that may be further up the intestinal tract.
Thank you, Mark. Kate, you have really fascinating data who complement a lot of things which were discussed today. You were focused on PSC IBD. Do you have data how the gut-homing T cells are in NASH, NAFLD, or maybe even hepatitis C?
So I guess from the data that I showed, I didn't have any hepatitis C. I had NASH, PBC, alcoholic liver disease, and there were similar levels of CCR9, alpha-4 beta-7. And I think that in the inflamed liver, you have upregulation of VCAM1, which alpha-4 beta-7 is a ligand for alpha-4 beta-7 as well. So I think that the inflamed liver attracts these gut-homing T cells. And I think it's irrespective of whether it's PSC or another inflammatory liver disease. But if I might offer something that can combine all of our talks together, and that is that I believe that PSC is an antigen-driven disease in an HLA primed individual, because there are very strong HLA associations from the genetic studies, and that having the right gut microbiome that then primes these T cells, and then the gut-homing T cells are expanding with the antigen exposed gut-homing T cells expand and go to the liver and cause their mischief. So that is potentially my hypothesis of how we can draw all of this together, whereas there's an interaction between these gut-homing T cells and specific gut microbiota.
Thank you, Kate. And there's a question from Jeff, who is online, and this is directed to Peter. The results are so dramatic that it would need about 20 patients in each arm for RCT. Is this correct? Is it worthwhile doing? Are you planning to do it?
Thank you for the question. The results are dramatic. And the power calculations are quite difficult because we're talking about a very heterogeneous group of patients. So we have done some power calculations. We have talked about randomized control trials, but we came up with a slightly bigger figure. So the two big for a single center such as ours, and certainly a multi-center trial will certainly be able to address it. Because the results are so striking so early, as in patients start to feel better very quickly. Within 6 months, they have weeks out of normal function, and 3 to 6 months out of normal scope. So it's a very easy study to do on the gut. The problems with the PSC, the multiple condition, the outcomes in the liver are far harder to quantify. Just improving LT, but we've also seen that can happen spontaneously in kids. So certainly in the gut point of view, we have certainly entertained and applied for funding for RCT. No, we haven't got anything that's fertile around.
Thank you. Kim, you really presented fascinating basic science data, and I think these animal models can be an important platform to develop therapies. Clinicians treating or dealing with PSC patients are always concerned about malignancies, and what we see in the liver or with the bile ducts is cholangiocarcinoma or colorectal cancer. HCC is not the biggest concern. Can you speculate a little bit why your animal models are more leaning towards the hepatocellular cancer and not towards the bile duct cancer? So is this really the right model, or is this just at a different stage?
Yeah, I think probably it's not the right model for looking at cancer from following PSC. I don't have the answer as to why it's developed a HCC, but I guess it's probably a better model of just an inflammation in the liver causing hepatocellular carcinoma rather than a PSC cancer model.
Other questions from the audience? Ranjeny.
Can Mark please explain how the bile acids and the vancomycin go together? Does the vancomycin somehow reduce the delivery of bile acids toxicity to the liver, or how is this fitting together? Because for me, standing back, this seems to be important. And also to the presentation of antigens, is the antigen going to be coming from a microorganism, or is it because of self-antigen that's released because of toxicity on a particular cell type? I'm just trying to put these bits and pieces together.
Yeah. Look, there are changes in bile acid profile associated with just IBD itself, and an increase in sulfated bile acids as compared to secondary bile acids, which are deemed to be the result of microbial activity. And it's the changes in the microbiota, the so-called dysbiosis with IBD, and the removal of the organisms that are responsible for those changes, that when they go back to the liver, they come back as sulfated bile salts instead. So I think the question becomes, and again, because I lack the clinical knowledge or expertise, to a certain extent, it's a question about what is the quantity and perhaps the profile of the bile acids that's entering at the early stages of PSC into the gastrointestinal tract, and is that exacerbating or accelerating the dysbiosis that takes place because of those different bile acids, or perhaps selects for bacteria that actually can use those sulfated forms. Something that happens that is overlooked a little bit is not just the loss of certain microbes, but the introduction or expansion of others that take advantage of those sulfated bile acids to meet their sulfur requirements, and that drives the changes that take place. So Ranjeny, for me, to a certain extent at this stage, the nexus between bile salts and bile salt metabolism changes in the microbiota and the, you know, disease course is still a bit chicken and the egg, like it is for a lot of the microbiota research. And I just don't know enough at this stage whether the onset of the damage that's taking place in the bile ducts and maybe the changes in the bile acid profile that's reaching the larger, the distal regions of the intestine is further driving the changes in the microbiota that then exacerbate complications like IBD. I think the issue, Kim brought this up, but you know, the mouse study shows that almost textbook kind of microbe association where you get a bloom in gram-negative bacteria, you get more LPS reaching the bloodstream because of translocation of bacteria and LPS, and in the liver then a TLR4 response. And there can be ultimately, in that study I think that you referred to, they showed myeloid-derived suppressor cells were a key suppressor of, you know, the anti-tumor immunity, and as a result, the onset of cancer. And that's probably what they see in the animal model. But it doesn't link well, I think, with what microbiota changes are taking place with PSC. That's all gram-negative, whereas the vancomycin effect and the changes that we're seeing, you know, more related to gram-positive bacteria.
Maybe for those who are not very familiar with the bile acid metabolism, we secrete primary bile acids into the lumen of the gut. They are metabolized by the bacteria, and interestingly, the secondary bile acids are then reabsorbed, and they're reabsorbed while they transit, and they are reabsorbed in the colon. And the interesting thing is we have the inflammation on the distal terminal ileum, right-sided colon, and frequently there's nothing on the left side, when we can assume that the bile acids are gone, they are reabsorbed. And my working hypothesis is that we have a toxic bile acid which recirculates, it's reabsorbed, and then damages after being reabsorbed and transported via the portal vein into the liver and excreted again. And this causes a problem. And the vancomycin, with a very narrow spectrum, potentially suppresses the activity of one bacterium which in genetically susceptible subjects produces this toxic bile acid or toxic substance, and this interrupts this circle. And I think this is where the microbiome and the very narrow spectrum of vancomycin nicely comes together, and this is how we can do it. Therefore, this would offer the opportunity to use something else to block this, suppress it, or replace such a microbe.
Yeah. I guess just very quickly to follow it up, I think potentially that's the real advantage of this combination of microbe culture with metagenomic sequencing. So rather than just trying to predict or find it through sequence gazing, you have the consortia of microorganisms and you can expose them to those types of bile acids and see if there is a specific bile acid type that's made and identify the organism that's responsible for it, or even treat that consortium with vancomycin and see if you change that profile. So by having the bugs in culture and being a holistic representation of what's actually there, gives you the opportunity to do bigger and better things.
There's another question online from Robert Anderson. Can you comment on the emergence of vancomycin resistancy as a negative aspect of long-term vancomycin? Peter, you looked at this because this is always a concern in the pediatric cohort.
Peter, we can't hear you, by the way. Just bring your microphone up.
I mean, maybe I repeat it then.
Oh, we can hear Gerald, just can't hear Peter.
Is that any better?
Yeah, that's
cool.
better.
Look, we have treated a number of patients over a number of years. We haven't had a single case of vancomycin-resistant enterococcus. It's a condition of going on vancomycin that patients have 3 monthly VRE either by stool or rectal swab. I can't say we've got 100% pick up 100% reliability every 3 months, but every patient who's been on vancomycin has at 3 months or 6 months and subsequently had their stool tested for VRE. We haven't had a single case of VRE or vancomycin resistant enterococcus.
And just to add to this, in the adult cohort, we have checked our patients as well, and so far not a single case. And Graham, a burning question.
This is a great meeting. Thanks very much to the organisers for the meeting. I've got a question. It's partially so, the problem with IBD is, doing PSCs and multiple subunits, and it's difficult, obviously, with numbers, not all as much in challenge PSCs and severe disorder. But in all the data that's been accumulated in the pediatric literature, are there any particular subunits, either in the bowel or the liver, that seem to respond more effectively to that?
That's a great question, and I was actually thinking about the time I spent coming to see you many years ago, and I knew you were going to ask this question because, well, no, phenotyping is so important to try and understand cohorts. Very early on when we got into IBD, we realised we weren't particularly. We made a lot of errors with phenotyping, and even now we're finding a lot of patients labelled as Crohn's disease actually have PSC colitis, labelled Crohn's disease because they had backwash ileitis. Phenotyping is very important. And now what we got data on about 60 patients who were given oral vancomycin to, those we absolutely know had PSC confirmed on MRCP and liver biopsy. Those that we've suspected PSC based on their phenotype and abnormal LFTs and p-ANCA positive, but without MRE or liver biopsy confirmation, and just the Hail Marys of very early onset IBD or patients who are just not responding to everything else. And it's quite striking that the ones with PSC have this incredible response. The Hail Marys for colitis don't. So there is something about that PSC finding on the biopsy or MRCP, but we've also had very good response, but not quite as good, where we've suspected PSC based on the phenotype. And again, someone's failing conventional therapy. Let's try some vancomycin. They've got a reverse gradient. They've had maybe some abnormal FTs, but we've not been able to demonstrate on their MRCP. We don't do a routine liver biopsy. We do a lot, but not routinely. And again, we've had a pretty good response to that. But again, that's a different story, and that's a harder story to sell because people aren't quite ready to believe that just because you've got a gradient, reverse gradient colitis, and you haven't yet developed PSC, well, you can't even really call yourself PSC-UC until you've clearly demonstrated it. But we've certainly had very good response in that right-sided reverse gradient p-ANCA positive ulcerative colitis. So in answer to your question, there's nothing else about the PSC on the biopsy which predicts it, but certainly p-ANCA positivity and PR3 positivity, which obviously tends to go with having the diagnosis of PSC, is quite a strong marker.
Thank you, Peter, and thank you for the question. I mean, my task is to wrap it up, and I think the most important conclusion for me is that the chair has miserably failed to keep the speakers in time. Ranjeny, you have a very last question?
Peter, so I'm going to ask you and Kate, is there a place potentially for failing T-regs therapy? Because there's quite a lot of trials now with lupus with that trial too. There is a very non-toxic lupus, advanced T-regs that absolutely is managing stuff. So is there a potential for doing that? And so Kate, could some of the CCL9 as well as actually T-regs and potentially efficient?
I think that question was for me, but I couldn't hear it properly. Gerald, if you could summarize it for me, please.
The question essentially was, are there Tregs, other immunosuppressive, immunomodulatory approaches which could make sense other than modulating the microbiome?
Yeah. So they're looking at that in Birmingham. They're doing a big focus on Treg cells and also looking at mate cells as well, actually. And they're looking at doing T cell therapy. They're starting off, they've been doing quite a lot of work with explants. So they basically have invented a lot of the protocols for isolating these T cells and doing it all. So they're very interested in Tregs. I didn't look at Tregs at all because you get a very limited number of cells out and you have to pick your panel. And depending on how good your flow cytometer was, I had a 12 color panel. So I didn't look at Tregs, but there's definitely work looking at immunotherapy with it. And I probably don't have much more to give you apart from that. And I think there's Christoph Schramm and his group in Germany are also looking at Tregs.
Okay, thank you. No further questions. Therefore, obviously, this is a hot topic. It's evolving. Our randomized prospective trial hopefully will shed some light on the mechanisms, maybe finding the bug which causes this. Then we don't need to talk any longer about immunosuppression, but really targeting the cause of the disease. And I'm optimistic that we will manage to do something like this because we now even see within non-related family members transmission of this. Therefore, there's some evidence that it's a transmissible disease. Therefore, just treating this transmissible factor could play a role. Therefore, stay tuned. It will be interesting. And I think Brisbane is a great place and the TRI is a great enabler for this kind of work. Kate, thank you very much for being patient with us. You stayed on time, and I look forward hearing more from your work, and I'm very confident that you will drive this in Adelaide. And thanks everybody who was here, contributed to his question and attention, and there should be time and opportunities to network outside and have a couple of stimulants for the gastrointestinal tract. Thank you very much.
Thank you. Bye.
All good.